Hello, everyone. My name is Man Gi-song. I'm Deputy Director General of Science of IVI. I will be moderating day 2 session. This is immunology of COVID-19, and the presenters are Hazel Dockrell and Nakon Premsri and Stanley Flokin and Peter Kwang. Before we get started, I would like to share a few housekeeping notes. First, our live session will begin at this time every day and this week, and the recording of each day will be made available on IVI's YouTube channel on the same day at 3 p.m. Eastern Time. These recordings will only be accessible during this week. Most of our lectures will be followed by a live Q&A. Please submit your questions in the Zoom Q&A and kindly understand that we may not be able to address all your questions live. Course observers will review and refer your questions to the lecture as time permits. Presentation slide files will be shared after the course. Short polls will be sent to you at the end of every lecture. These polls are anonymous, and your feedback on the lectures and speakers will be much appreciated as we develop future courses. For those who are joining us live on Zoom, a digital certificate will be available upon request. We'll note your Zoom login details, and attendance rate of 90% throughout the week will qualify you for certificate. Now let's please welcome our first presenter, Dr. Hazel Dockrell. Dr. Hazel always been a best lecturer of IVI Vaccinology course since she started the lecture several years ago in this course. Dr. Hazel, please.
Hello, I'm Hazel Dockrell, and I'm Professor of Immunology at the London School of Hygiene and Tropical Medicine. I work on tuberculosis immunology, and I've been privileged to come and talk to the IVI Vaccinology course about basic immunology for a number of years. So what I want to tell you about is something of what, as vaccinologists, you really need to know about basic immunology. Now, the immune system is facing just a huge range of challenges. We are exposed to pathogens from the smallest viruses to the largest helminth worms, so anything from submicroscopic to tapeworms that are meters long, and we're exposed by inhaling, having vectors inject, ingesting, and sexually transmitted infections as well. So a whole range of different routes. So the immune system is facing a lot of challenges, very large to very small pathogens coming into the body by different routes. And we obviously need a very complex and an adaptable way of defending the body from these infections. Now, the immune system can do that. And I think this is it's a very old example of measles epidemics in the Faroe Islands. Now, the Faroe Islands had 2 epidemics of measles, 65 years apart, and no measles in between. And after the second outbreak, the doctor who was investigating noticed that none of those who had been infected in the first epidemic were infected in the second outbreak. So basically, there was immunity in those that had been infected the first time round, and it lasted 65 years. So the immunity was specific and long-lasting. And it's this ability to actually induce and maintain immunological memory that vaccines exploit. And we're very lucky that vaccines do, and we've had this tremendous success in the last year, where vaccines to COVID-19 are doing better than natural infection because they are protecting individuals from not only becoming infected, but from becoming very ill. They're doing it in a safe and controlled way, and they are actually inducing long-lived immunity. We've had plenty of other examples of vaccines that do that before, and this slide shows some, for example, polio at the top, measles, rubella, and mumps, where the vaccines were introduced and there was a dramatic drop in the number of cases of the disease. But the question for immunologists and for you is, how do the vaccines manage to do this? Well, they do it by inducing active immunity. When we're exposed to the microbial antigen of the infection or the vaccine the first time round, we make an immune response, and that immune response is then able to fight off and defend us from the infection. If we then are challenged by a second infection with the same organism, we are then immune. We have a specific immune memory specific to that infection, which is long-lived. And that's different from passive immunity. Passive immunity is, for example, when you take serum or antibodies from somebody who has recovered from infection and you give it to somebody else. And those antibodies will give you temporary protection and allow someone to recover. But it will not give you memory, because those antibodies will then be used up and disappear, and you'll be left unprotected. But the way in which the immune system does this is very complicated. We have different immune organs. We have primary immune organs like the thymus, where T cells develop, and the bone marrow, where many of the immune cells develop. We have secondary lymphoid organs like tonsils and lymph nodes and spleen, where many of the immune responses are initiated. And then we have the system of lymphatics that will help circulate the cells around the body. Then there are a whole range of different cell types. And on those cells, a whole myriad of different immune markers and receptors on the surface of the cells, and then signaling molecules and cascades within the cell that will then instruct the cell to make the immune response that is going to be protective. So that is incredibly complicated, and I don't think you need to know the fine detail of that. What I think you do need to know is that immunity is why vaccines work, that vaccines can induce specific and long-lasting immune responses, and they do this, they're based on very precise recognition and binding mechanisms. And I'm going to tell you a bit more now about T and B cells or lymphocytes that express these specific antigen receptors on their surface membranes. What happens then is that the specific cells are increased in number, and they are primed so that they will react more quickly and effectively when we meet the same infection again. Those immune responses use there are specific immune responses. They use non-specific mediators and cells often to then carry out the killing or the removal of the pathogen. And then there needs to be cooperation between both what we call non-specific and specific adaptive immunity, and between these various cell types. So there is a system of what we call innate immunity that is not specific in the same way that the adaptive immune system is, but it has, for example, barriers to infection like epithelial barriers. It has cells like phagocytes, like polymorphs, like monocytes and macrophages, dendritic cells, which we'll hear more about in a minute, as they can present antigens to the T and B cells, natural killer cells. And these cells give us some temporary protection while the adaptive immune system becomes activated. The B cells and T cells in the adaptive immune system are then activated by their specific antigens. They proliferate, and then they become effector cells, either making antibodies for B cells or becoming effector T cells. Another thing just to note at this point is you will be hearing more about adjuvants in vaccines, and adjuvants often induce an inflammatory response in the cells of the innate immune system. So the B cells make antibodies, and I think you all know about antibodies. Each B cell has antibodies of one particular specificity on their surface, and that acts as their antigen receptor. And the specificity of the antibody is in the antigen binding site right at the end of the molecule. B cells also then make and secrete these antibodies as soluble antibodies, which then circulate in the serum and gives protection. There are different types of antibody, different classes, and even subclasses of antibody. And how well they work will depend on how strongly they bind to the antigen. The ones that probably are most important for you are the IgG antibodies, which will carry out most of the neutralization and opsonization or binding to the microbes, and then IgA antibodies that will give us protection in the mucosa. So those antibodies can carry out a whole range of different functions. They can neutralize toxins and microorganisms. They can opsonize for phagocytosis. They can help, for example, natural killer cells kill an infected cell. And they can work together with components of the innate system, like complement components, again, to phagocytose, to induce inflammation, and to lyse microorganisms. T cells are more interesting and more complicated. I'm actually a T cell immunologist by nature, and whereas the B cells secrete antibodies that are really designed to see antigens in solution extracellularly, the T cells are only seeing antigens displayed on the surface of the cells. And they are seeing those cells presented by either the HLA or what we call MHC, major histocompatibility complex antigens. And it depends where the actual antigen is in the cell, what sort of presentation pathway those antigens get into. If in the top half you have a virus that gets into the cytoplasm or the cytosol, those antigens are presented to a CD8 T cell. So viruses like SARS-CoV-2 can activate CD8 T cells. If they are in organisms that are phagocytosed, like M. tuberculosis, for example, they will then get presented by MHC class 2 molecules and activate CD4 T cells. So there's an initial repertoire of T and B cells, and they have specific and diverse antigen receptors on their surfaces. And the way the immune system works is that when you meet an antigen, then that particular specific lymphocyte will be activated, will then proliferate, so that you'll get clonal expansion and more of those cells, and those cells will then differentiate to become effector cells. It gets more complicated than that because those effector cells are also specialized. I told you a little bit about how B cells and their antibodies can carry out many roles. T cells also can carry out different roles. The CD4 T cells will carry out roles including activating macrophages, inducing inflammation, and also helping to activate and stimulate other T and B cells to proliferate. But then the cytotoxic CD8 T cell will be able to kill, for example, a virus-infected cell. And then there have to be some regulatory cells that will control the whole system from getting out of hand. But the bottom line here is that vaccines need to induce both B and T cell immunity. Even the CD4 T helper cells have got subdivisions within them that make different groups of cytokines. So, for example, for M. tuberculosis, which is one of the examples I'm using, they make interferon gamma to activate the macrophages. For helminth infections, for example, they make another group of cytokines like IL-4 and IL-5. So the lymphocytes are themselves facing challenges. You start with very few of them, specific for any one microbe, and so you have to expand them by proliferation. They must be able to locate and respond and get to the right place in the body by migration. And then they need to specialize to effector cells. So vaccination is able to induce specificity and memory. And the memory will induce more cells that are easier to stimulate and that are already differentiated. And here you can see a primary immune response in purple to antigen X, inducing B cells making antibodies, plasma cells, memory B cells. But then when you meet that antigen again, you make a secondary immune response, which is much larger, quicker, and more effective. And that is the whole principle of prime boost regimens of vaccination, where you give 2 or more doses. So memory B cells and T cells provide long-lived immunity. There are long-lived and there are long-lived B memory cells and long-lived T central memory cells that are circulating and are held in reserve for when we meet an organism again. There is also an interesting phenomenon that I haven't time to talk to you about in detail, but there is some hint of memory in the innate immune response, both in macrophages that seem to have some epigenetic changes in the way the genes are primed to respond, which is called trained immunity, and probably something similar going on in natural killer cells. That was the basis of people trying to use BCG in trials to see if it would provide some initial protection against SARS-CoV-2. So the phases of adaptive immunity are to induce these specific cells to expand, to differentiate, to carry out their effector roles. And then you do have to reduce the number of those cells, because otherwise you just don't have enough space in the body to keep them all hanging about until you need them again. So most of them die by apoptosis, but you end up, or programmed cell death, but you end up with surviving memory cells. So, for example, for SARS-CoV-2, it's the spike protein that is in most of the vaccines, and you induce antibodies to that spike protein. That spike protein is critical because it stops the virus from binding to the receptor on the surface of the host cell, the ACE2 receptor. But following vaccination, you do get, although you get both T cells and B cells to vaccines that are made using either messenger RNA or delivering the genetic material through an adenovirus in the AstraZeneca vaccine, both vaccines in the body will make the SARS-CoV-2 protein, spike protein, and induce both specific T cells and B cells. But those antibodies will then fall away after vaccination. And this shows, for example, particularly clearly for the Pfizer vaccine in the blue top bars, how antibodies decrease after vaccination. You can see the antibodies are decreasing with time, but the good news is that they can then be boosted by when you are exposed to SARS-CoV-2 again. Most of the successful vaccines that we have, including SARS-CoV-2, are vaccines that induce antibodies, and those do seem much easier to develop successful vaccines for. There are some at the bottom, HIV, malaria, and TB, which have been more difficult. And I think the reason for that is, for example, if you take M. tuberculosis as an example, it induces a more complicated immune response because you inhale the organism, it then lives in the macrophages. Some of its antigens are picked up by the dendritic cells and presented to T cells. You do induce and activate CD4 T cells, making a range of cytokines. You are able to wall off the infection in a granuloma, but often you're not able to completely clear the infection. And it can then develop later into a progressive TB infection. And actually, despite much effort, we haven't yet got good correlates of protection for TB. We know that CD4 T cells are important. We know that interferon gamma is important, maybe even antibodies, possibly CD8 T cells. So the immune system, it can recognize self and non-self. It can communicate. The cells can communicate with each other either by cell-to-cell contact or by these soluble mediators called chemokines. They can migrate, getting to where they're needed. They can block infection, either with antibodies or by killing the pathogen within host cells, specialize to make the most appropriate type of immune response, and then respond faster and better when re-exposed to the antigen. And it's that memory response that is exploited by vaccination. Thank you.
Thank you very much, Dr. Hazel. You are already the best. I have 4 questions from the audience. The first question is, Is the level of immunity compared after SARS-CoV-2 infection similar in mild, moderate, or severe type of COVID infection?
Okay, well, now you're getting to good morning or good afternoon to start. First thing to say is I am not an expert on SARS-CoV-2, and you will have some more lectures from people who are more expert than me following on in the course. You need to have a certain degree of infection to induce a good immune response. The problem is if you have too severe an infection, then you induce a lot of immunopathology, and that really isn't necessarily going to give you a much stronger immune response. But I think that this is the sort of area that we're still beginning to try to understand the full detail of the responses to SARS-CoV-2. Thank you.
Thank you very much. Second question is, what is the difference between natural infection and vaccine? Because I think they are working with the same mechanism, or how can the vaccine has an advantage? Infection and vaccine, yeah.
Yeah, well, you're right. Both a natural infection and vaccination can induce similar immune responses. The vaccine can, for example, focus the immune response. And for example, for the SARS-CoV-2 vaccines, they are almost all focusing the immune response by forcing the body to make antibodies just to the spike protein to get very good levels of antibody to the spike protein. And that's because those antibodies will block the virus from binding to the ACE2 receptor and then infecting cells. Secondly, you can also induce a stronger immune response by the way that the vaccine gives the antigen. So many vaccines are using adjuvants, and those actually induce a state of mild inflammation. And you often know that when you've had a vaccine, you get a sore arm and sometimes you have a temperature, small fever for a day or so. And that immune response does seem to help push the immune system to make a better specific immune response afterwards.
Thank you. And the third question is, how do the single dose of SARS-CoV-2 vaccine work if they don't need booster primer? Can they also trigger memory immune response? The single vaccine, do you think enough memory immune response?
Yes, they do induce a memory immune response. I think that is due to a combination of clever design and the exact way in which the vaccine is made so that it will make an immune response with just one dose that is equivalent to giving 2 doses of some of the other vaccines. Again, I think you will probably hear a bit more about that as the course goes on.
Thank you. And this is a last question. What could be the outcome of administering 2 different vaccines in terms of immunological memory and protection against COVID? I think this is a mix and match.
Yeah, well, if you were giving 2 different SARS-CoV-2 vaccines, for example, one is the prime and the second is the boost, I think the evidence so far is that that is very effective and it does boost the specific immune response to the SARS-CoV-2 antigen spike protein, and that is very effective. If the question is slightly different, for example, in the UK at the moment, there has been some debate whether we will give a booster vaccine of SARS-CoV-2 and how we will administer that when we also should have a flu vaccination campaign. So some people are worried if you give 2 vaccines at once, will it inhibit or reduce the immune response to one of them? For example, you could give the flu vaccine in one arm and the SARS-CoV-2 booster in the other arm. As far as I know, that seems still to work. And people often say that, for example, the smallpox vaccine was very effective. And smallpox is
a virus that has a very large number of proteins, something over 200, I think, proteins. So actually, the immune system can cope with being exposed to a lot of antigens at once. And it is dealing with them, antigen by antigen, with them activating the specific T and B cells.
Thank you very much. Your great talk and answer questions. I think I hope to see you soon next year in IVI. Thank you.
I hope so. Yeah. I hope so too. And congratulations on running the course again. And I hope it's as successful this year as always.
Yeah. Bye.
Thank you. Bye-bye.
Now let's please welcome our second speaker, Dr. Nakon Premsley from Thailand National Vaccine Institute. Dr. Nakon, please.
So good morning, good afternoon, and good evening to everyone for the participant to the International Vaccinology course of IVI. I'm pleased to be a speaker today. My name is Nakon Premsley, a director of National Vaccine Institute of Thailand. I can say that I was the participant of this course as well. So welcome all of you to my talk today. So it will be about of the immunology, focus on the COVID-19 or SARS-CoV-2 virus infection. So it will be on the new information that we can learn on the dynamic of the pandemic of the disease for today. So let me start with my slide. So the talk of today is about the immunology, wild type infection, reinfection, and herd immunity. So the topic in about the introduction of the infection, the immune response to virus infection, as well as wild type infection. And I will touch on the variant of concern or variant of interest infection, as well as a reinfection of SARS-CoV-2 and herd immunity for your food of thought today. So when we say the immunology, it's about the study of the response of the body when it has been challenged by the antigens or the microorganism. And it's also called as the state of being resistant to injury or infection or poison or invading of pathogens. So it's a natural response of our body that when we see the foreign antigen or microorganism as a virus, protein, or bacteria or parasites. And immunity is in a kind of widely divided into two general types, that what we call the innate immunity or innate immune response, or adaptive immune response. So the immune response to viral infection, normally it will start with our first mechanism of the innate immune response. So it will defend for any microorganism or antigen that come into the body, and it's presented from birth and always available to protect us against the pathogen attacks or the disease. And its general response to any infection or foreign body to our body. And the innate immune response cells that can secrete interferon or the chemokines or what we call cytokines that interfere the bacteria or viral replication. So the cell that involved in the innate immunity is a macrophage, neutrophil, natural killer cell, or the dendritic cell. And the immune response to the viral infection, I would like to use the example from SARS-CoV-2 infection, that we also have the innate response that the dendritic cell or the macrophage cells, it will be the what we call antigen presenting cell. So when the microorganism invade into our body like SARS-CoV-2, And they use the spike protein to attach to our ACE2 receptor, and then it fills into our cytoplasm cell, and then the virus enter into the epithelium and fills with the vesicle at the mRNA that release, and then the virus assembling their self and get out from the cell and make the cell in apoptosis or a dead cell. So that when our innate immunity or dendritic cell that detect the viral antigen or viral particle, it will ingest or phagocyte that virus, and then it will come into the presenting of the part of the virus or the viral peptide to our T cell system, and then the T cell system will be stimulated, and then it will come in the two pathway of T cell and B cell at a cytotoxic T cell for generating of cellular immunity, and the B cell will produce the antibody, that what we call the humoral immunity. And regarding the stimulation of this, we will get the neutralization or the binding antibody occurring in our cell, and then we also have the cellular immune response that can generate the memory B cell or memory T cell that can be used in the long term of protection. So these are the mechanism of the working of the T cell and B cell. So the T cell recognizes a cell infected with a specific virus, so what we call the cellular immune response, and rapidly replicate to tackle the infection cell. And the CD4 cell or T helper cell will stimulate the B cell to produce antibody, and as well as the CD8, a cytotoxic cell, to kill the infected cell. And the B cell will make up around 15 to 30% of the lymphocytes in the body, and most of the B cells differentiate into the plasma cell, and that protect the microorganism or the disease by producing antibody. And it will be specific to the microorganism or virus. So the IgM antibody will be produced the first time and may disappear after a few weeks. But the long-term antibody, what we call IgG, that are produced at the same time of a couple of days later, remain for months or years, depending on the disease or the specific immune response. So I think that's the response to the natural infection or the wild type or the viral infection, especially SARS-CoV-2. But right now it's the typical of the virus that, especially the mRNA virus like SARS-CoV-2, that they can evolve over the time through the mutation, and then they change the genetic sequence. So if the changing is specific to the major point or the important point, like the spike protein that right now we recognize it is a kind of the major protein that will produce the antibody, and it's a major protein that attach to our cell, to our receptor. So a change of virus, what we call variants, can differ from one mutation to many changes of the spike protein, and possibly result in the virus transmissibility and diseasability. So right now we have several variants occurring, like naming by WHO using the Greek alphabet. So we can heard from alpha, beta, gamma, delta, and as a kind of variant of concern. And the variant of interest, it's the other virus that it's kind of in a way that we keep monitor on their or the mutation of the virus that can increase our transmissibility or the disease severity. So right now we have variants of interest or VOI or variants of concerns. And the variant of concerns that appear to be more infectious or cause more severe disease in other circulating coronavirus. And it's associated with the increase of transmissibility and increase of virulence or change in clinical disease presentation and effect to available diagnostics, vaccines, or therapeutics. It's assessed to be the variant of concern by the WHO in the consultation with the WHO SARS-CoV-2 virus evolution working group. And you can see the naming of the WHO used as a variant of concern that we have heard several times of alpha, beta, gamma, and delta, which was previously naming by the country that discovered the new variant, but to avoid of labeling and or stigmatization. So we appreciate the WHO label of the variants by using the Greek alphabet. And so it's caused a major thing as well, even by the new variant, because it will cause a reinfection of SARS-CoV-2. It's a kind of major factor that cause, because reinfection occur in the individuals who have recovered from prior COVID-19 infection because of the new variant or waning of immunity. And there is also an associated factor in the host factor like asthma or nicotine dependence, or the unvaccinated people can have more than 2 times that likely to get reinfected compared with the post-infection who have got fully vaccinated. So this is another of recommendation that even the post-infection, they should get a kind of vaccination to prevent of reinfection, especially in the situation of circulating new variants. And the infection rate it's so minimized, but it can cause severe illness. And the average infection occur in around 2.5 months after the prior infection. So this is the paper that we summarize from the study that analyzed from more than 9,000 patients in United States, and reinfection was identified around 0.7% of the post-infection individual. And another factor, part of variant, is a reduction of protection to the immunity. So this is an evidence that the neutralization sensitivity of the convalescent sera against SARS-CoV-2 variant is reducing or declining. So this is most of the sera significantly less effective to neutralize the variant and the wild type. This is an evidence that we see that N501Y that was the first variant that we observed. So it's significantly reduction of the neutralizing antibody titer to the variant comparing to the wild type. So this is the evidence that the variants that can cause reinfection because of the reduction of protection to the immunity. And by the collective of the information that the reduction fold of the neutralization antibodies against the variants that comparing or to the reference to the wild type strain or the early mutation strain as the 614G as a reference. So we can see a number of vaccine over here, or mRNA vaccine, viral vector vaccine, or inactivated vaccine, or even subunit protein. That by the study against the variety of variants, so we can see the reduction fold comparing to the wild type virus. We can look at the mRNA vaccine that when facing with Kappa or Delta, which is a very close variant as a Delta variant, the reduction fold for the mRNA is 6 to 7 or even 12 folds of reduction. And the viral vector, when against with the beta variant, the reduction fold is 6 to 9 of reduction of neutralization antibody, and the inactivated also reduction of the fold of itself. But for the inactivated vaccine, the level of immunity is lower than the other platform. So even it's a reduction for 2 or 3 times comparing to the wild type, but it can cause a breakthrough infection among the vaccinated people. But the subunit protein, it's less than the others. So this is the kind of information comparing between the platform, the vaccine, and comparing with the wild type neutralization. So this is the Delta strain. It's resistant to the therapeutic monoclonal antibodies and the natural infection or vaccine-induced antibodies. And it caused hyperlocal outbreaks and among the unvaccinated people, or even the post-infection people with the prior wild type strain can be at risk of a Delta infection. And it can cause more vaccine breakthrough cases that we can see in many countries right now. Even they have a high coverage of vaccination among the population. But even in the United States or United Kingdom or Israel, with higher vaccination coverage, but we can see more and more vaccine breakthrough cases. And the Delta variant, that what we know as B1617.2, it's almost twice as transmissible as the original Wuhan strain. And the US CDC has estimated that it's about 2 times that cause transmissibility. But it's not only more contagious, but make people more sicker. That we can see the study in Canada or Singapore or Scotland, that the higher risk of the pneumonia, hospitalization, or ICU admission, and or the oxygen requiring. So it's not just a breakthrough infection, but it can cause more severe the symptom. And the neutralization antibody activity against the Delta variant is our major concern. So we can see that it can cause more reducing trend comparing with the other variants. So it was 5 to 6-fold reduction of the neutralization, and the neutralization antibody reduced over the time. So when the people waning of the immunity and facing with the Delta variant, we will have more opportunity or more probability to get the infection. And again, it will be more severe than the previous infection. But however, the vaccine will remain to protect that even the lower of neutralization, but the 2 doses of the vaccines to be effective to prevent hospitalization and death. And vaccination is associated with the faster decline of viral RNA load, meaning that it will reduce the transmissibility of the virus as well. And the vaccines to be a key strategy to control of COVID-19 pandemic. So 2 doses of the vaccine can save the people from severe and deaths. And again, this is a declining of neutralization antibody against SARS-CoV-2 among the convalescents. So this is a waning immunity of the convalescent patient in the longitudinal study of the Wuhan hospital in 1 year cohort. This is another interesting information that we can see the declining trend of the antibody. And then this is one of the study that the effectiveness of Pfizer COVID-19 vaccine steadily declined over the time and dropped to about 84% for vaccinated people after 6 months. And then there will be more chance to get infection even fully vaccinated. So found that it was strong in the initial time, but it declined in average of 6% every 2 months. And you can see that by the over 4 months after 2 doses over here, the vaccine efficacy declining from 96 to 83% of protection. And in the Israel study, that Pfizer shot is just 39% effective after 6 months. So this is the dynamic information that we need to monitor. This is another evidence on the elapsed time of if the time more than 146 days, the chance of getting infection is more than double in every age group, even post-vaccination of mRNA vaccine, especially among the one who age over 60 years old. So this is an evidence of a declining and a breakthrough infection. So what's about the herd immunity? Because it's known as a population or community immunity that provide the indirect protection effect from the infectious disease for the unvaccinated people if we have adequate vaccine coverage. So it's through the vaccination, or it can be developed after the natural infection. So it could occur when a larger portion of the population becomes immune by the disease or vaccine, and the whole community can be protected. So this is a theory of herd immunity. And to achieve the herd immunity against COVID-19, we need a substantial proportion of coverage of vaccinated population. And herd immunity is only relevant if there is a kind of transmission blocking vaccine, or meaning that the vaccine that can prevent infection, not just symptomatic disease or severe disease, but we do need the vaccine that can prevent infection or asymptomatic infection. And it would need a kind of asymmetrical or even vaccine rollout throughout the world, that we all throughout the country, so that we can get close to the herd immunity level. But however, the new variants and the waning immunity that I have said previously, we have seen over time is very challenging to reach the level of herd immunity. And individuals, regardless of the history of prior infection, should continue to practicing social distancing and mask wearing. So this is the picture of explaining of herd immunity. So if we have the people more vaccinated, so we can prevent the infection for the others who has not been vaccinated. So the blue color for the non-immunized people, and the yellow color for the immunized people, and the red color for the sick or for the patient. So by the picture, if we have more and more vaccine coverage by more and more yellow color of the people in the community, we will have less opportunity to get infection. So this is the picture of herd immunity. However, what we have seen for now, the herd immunity, depending on the what we call R0 or R0, that calculated from the coverage of the vaccine, that should be have a good coverage of the vaccine. Previously, COVID-19 vaccine would need around 75 coverage of the population to reach the herd immunity by the estimated of R0 about 4. But now it has been changed by the epidemic of the variants. So if by the calculation or by the study of the US CDC that saying that it will be more than 2 times transmissibility, so the R0 maybe reach to 8 to 10, that can close to measles that we can see in the table, in the middle table over here. If it come close to diphtheria or measles, so we would need a higher coverage of the vaccination. So it may be around 90% of the population needs to be vaccinated rather than 75%, meaning that we would need more vaccine for the people to stop off the pandemic. So the potentiality to get new variants, so we will not face only Delta variant, so we can expect to see more and more variants because the pandemic still be going on, and the virus replication can cause a mutation. So the new variant can occur. So to minimize the potential of new variant, we do need a kind of equal distribution and fast distribution of the vaccine all over the world to stop the pandemic. So that's why the WHO is saying that no one is safe until everyone is safe. So if we still have the potential of the area that can cause a huge outbreak, like in India or in South Africa or in Brazil, so we can expect more and more variants, and the vaccine needs to develop as kind of run after the variant occur. So even right now, we don't have the second generation of the vaccine. Even the investigator or the scientist is keeping a study on the second generation to address the new variant. So this is the way that we need to protect ourselves from the COVID-19. The control measures still need to be emphasized and practiced by the distancing, hand washing, and masking. So I would stop here for the presentation, and happy to address any question and answer after this. Thank you very much.
Thank you very much, Nakhon, for your wonderful talk. We already have several questions for you. I think it's 3 questions now. So the first question is, if the booster shot is needed, how is more effective between homologous or heterologous boosting? What is your opinion?
But thanks for the question. I think it's come to more and more evidence on the heterologous booster dose that we can see from the several studies right now, that by the different platform of vaccine may provide a good immune response. However, we also see the booster effect from using the same platform of the primary series, like we see booster dose of a booster effect from Sinovac vaccine inactivated by the third dose. And we also see the third dose effect from AstraZeneca. I mean, by the same platform or homologous booster. To me, I think we may not we may need not to use only an antibody level, because the other factor is about the cellular immunity, which we need to take into account as well for a kind of longer term of immune response. Because if we look at only the antibody level, it would be a kind of only one part, because our system is in both systems, cellular response and immune and humoral response. So I cannot give the best answer for now, because we have seen the booster effect from both the homologous booster and heterologous booster.
Thank you very much. And second question is, what could be the possible reason for the reinfection after the vaccination? I think it's some immune responses, or can you explain that?
Do you mean the reason why we have a reinfection?
Reinfection after the vaccination, yeah.
Right. I think it has said in the presentation two factors. It's about the waning of immunity and the variant of the virus. So we need to realize, because the vaccine has been developed in a very urgent manner, and we haven't got the kind of long-term effect of the vaccine. So it's a kind of an ongoing. So we did not have a good evidence of how long would it take for the protection period. But it seems to be more and more evidence that even after prior infection or after fully vaccinated, we can see the reinfection in around a 6-month period after that. So both factors, waning of immunity and the circulating of the new variant, it's a reason of the reinfection, apart from a host factor. But host factor it cannot predictable. Some of individual may get a higher immune response or a longer immune response, depending on host factor. That I could not say in specific. But for the other is the waning and circulating of variant.
The last question is, I think, can you assume that herd immunity is not reachable with our current tools, given that the vaccines are not transmission blocking, especially in the context of variant emergence? Do you have any ideas about this?
Yeah, for this moment, we need to keep eyes on this. But the hope of herd immunity is a little bit far by two factors, and of the mutation of virus, the new variant, and escape from immunity. Even we have fully vaccinated. And the second one is about the efficacy of the vaccine on blocking of transmission. Because for the moment, we can measure the effect or the effectiveness of the vaccine by only prevent of symptomatic infection. But we right now we have not yet established the kind of prevent of infection effect from the vaccine. Even we have seen some of an evidence of reducing of viral load in the vaccinated people, meaning that it can reduce transmission, but it's not 100% blocking of transmission. So we can see the transmission by the vaccinated people to the unvaccinated people. That's why herd immunity is a bit it's a bit far by the current time, unless we can get a better vaccine, or we can mix and match of the vaccine regimen, that we can see the higher effect or the better effect of the vaccine after this.
Thank you very much for comprehensive opinions. Thank Dr. Nakon for your great talks today. Yeah.
Thank you very much. Sawasdee krab.
Now our third speaker. It's a great honor to me to introduce Dr. Stanley Flokin. Dr. Stanley Flokin is Emeritus Professor of the University of Pennsylvania. He is now a consultant to vaccine manufacturers and non-profit research organizations. He is a member of Institute of Medical Medicines of the National Academy of Science and the French Academy of Medicine. Actually, he published more than 800 articles, and he has edited several books, including a textbook on vaccines. He developed the rubella vaccine, which is now in standard use globally, and he also co-developed the pentavalent rotavirus vaccine. And he worked extensively on the development of application of other vaccines, including anthrax, oral polio, rabies, varicella, and cytomegalovirus. So today he'll talk about correlate of vaccine-induced immunity. Dr. Stanley, please.
I'm Stanley Plotkin, professor at the University of Pennsylvania, and I'm going to speak on correlates of vaccine-induced immunity. Next slide, please. So why are correlates of immunity important? Because, of course, we want to know basic immunology and understand it, but also correlates enable the correct choice of a vaccine antigen. It permits consistency of potency from one lot of vaccine to another. It determines the susceptibility of an individual or a population. And if an efficacy trial is not feasible, immunologic data may enable the licensure of a vaccine. And finally, it enables bridging from first generation to second generation vaccine. Next slide. Now, terminology is important in this area. So a correlate of protection is defined as an immune response that is statistically interrelated with protection. And it may be an absolute correlate, that is a threshold. It may be a relative correlate, that is a correlate that is variably correlated with protection. And there may be more than one correlate. Next slide. And one point that's important is that there are mechanistic correlates of protection, which is defined as an immune response that is actually responsible, biologically responsible for the protection. Next slide. On the other hand, there are non-mechanistic correlates, which are immune responses that are substitutes for the true immunologic correlate, which may be unknown or not easily measurable. An example of that is correlate of protection against zoster. We know that cellular immune responses are actually the true mechanistic correlate, but we use antibody to varicella virus to determine whether a zoster vaccine is working, because it's much easier to measure antibody. Next slide, please. Now, I'm not going to read this slide, but just to point out that there are many immune mechanisms that are induced by vaccines and may be correlates of protection. But we are going to try to simplify as much as possible by identifying the correlates that we know for certain diseases. Next slide. So the first principle is we must define protection against what? Infection or disease, for example. In the case of polio, for example, the infection is prevented by antibodies, and also the disease is prevented by antibodies. But it's much easier to prevent paralysis from polio by antibodies in the bloodstream than preventing infection, which requires antibody in the intestine. Next slide, please. Another important idea is that correlates of protection may vary according to the clinical presentation. For example, the pneumococcal conjugate vaccines prevent pneumococcal disease, but it requires much lower titers to prevent bacteremia, as shown in this slide, than to prevent otitis, for example, or nasopharyngeal carriage, so that correlates of protection may vary according to the disease that we're trying to prevent. Next slide. The mechanism of protection against infection or vaccination is not necessarily the same mechanism as recovery from infection. So that, for example, next slide, in the case of measles, we know that antibodies can prevent infection. And as this slide shows, that it can prevent infection. And actually, if the antibodies are high enough, it can prevent disease and infection. However, in order to recover from measles, if you are infected, you require cellular immunity. And that's why individuals who lack cellular immunity may have a very severe measles. Next slide, please. So this is what I've just said, that B-cell deficient humans recover from measles, whereas T-cell deficient humans may suffer serious and fatal disease. Next slide. So a third principle is that a large challenge dose can overcome immunity. And this has been demonstrated, for example, with oral polio vaccine as a substitute for a wild polio virus. And you could show with oral vaccine that if you give a large enough dose, you can overcome pre-existing immunity, whereas you can prevent a small challenge dose of the oral polio vaccine with lower antibodies. Next slide. And indeed, most current vaccines do protect through antibodies. And that's because, next slide, most of the diseases for which we have vaccines act through viremia, bacteremia, or toxin production, and all of those are prevented by antibodies. So again, most of the diseases for which we have correlates protection are antibodies. Next slide. A fifth principle is that correlates may be relative. Next slide. And by that I mean that severe disease may be prevented by a lower level of antibody than mild disease. And a good example is pertussis. As shown in this slide, whether you're dealing with household infection or non-household infection, lower levels of antibody may not prevent severe disease if an individual is infected, but higher levels will make the infection mild or may completely prevent the infection. Next slide. Another principle is that antibodies must be functional. Next slide. And this is an example from meningococcal polysaccharide vaccination, a trial that was done in Quebec in Canada. And the results of that mass vaccination were analyzed according to the age of the vaccinees and the presence, the induction of ELISA antibodies or bactericidal antibodies. And what was demonstrated that young children could develop ELISA antibodies, but it didn't necessarily protect them. It was only those who developed functional bactericidal antibodies that were protected. So infants only had 18% efficacy, or even lower than that, 0% efficacy, but adults had 83% efficacy. So in this case, the level of bactericidal antibody was critical. Next slide, please. So another principle is that there may be more than one factor that protects as co-correlates. Next slide. An example of this is influenza. Using the live intranasal influenza vaccine as an artificial challenge. And in this study, it was demonstrated that children who had pre-existing antibodies in the serum and in the nose had very low infection rates. So they were very well protected against influenza. But children who had only serum antibodies or only nasal antibodies, shown as the 19% and 15%, could be infected to some degree. Of course, those who had neither antibodies were highly susceptible to infection. But so nasal antibodies and serum antibodies are co-correlates for influenza. Next slide. Another principle is that memory may be a mechanistic correlate. And the example of that is hepatitis B vaccine, which disappears, that is, antibodies disappear with age, but people are still protected because they have immune memory against hepatitis B. Next slide shows that T-cell responses can be correlates. And the example of that is tuberculosis. Next slide. And the illustration here is bovine tuberculosis, and the measurement of cellular responses, T lymphocyte responses in cows showed that that only cows who had T-cell responses to tuberculosis were protected, whereas those that didn't would be susceptible to tuberculosis. Next slide. So this is a slide on the correlates of protection against intestinal pathogens. And I'm not going to go into detail, but just to say that correlates against intestinal pathogens are complex because it's not only the presence of innate immune responses, but also mucosal secretory antibody, mucosal IgG antibody, and yes, even serum IgG antibody and T-cell responses may influence infection and protection against intestinal pathogens. Next slide. Now, I'm going to say a few words in closing about correlates of protection against the SARS-2 virus, the cause of COVID-19. So this is a cartoon of the virus. And as everybody by this time knows, on the outside of the virus is the spike glycoprotein. And the importance of that is that neutralizing epitopes, especially in the receptor binding domain of SARS-2 virus, are inducers of neutralizing antibodies. And most of the vaccines that we have use the spike protein to induce neutralizing antibodies. But I do want to mention that there are other antigens like the membrane glycoprotein and the envelope glycoprotein that could be used in vaccines to give breadth of response. And that's something for the future. Next slide. Shows that, as you know, there are many different vaccines against SARS-2, from inactivated vaccines to live vaccines to vectored vaccines. Next slide. And yes, there are protein subunit vaccines and, of course, nucleic acid vaccines, both DNA and RNA vaccines, which have been successfully used. But I want to focus on the correlate of protection, which we have learned about. Next slide. So this is a diagram that shows two things. Well, first of all, it shows 5 different vaccines and the immune responses in terms of neutralizing antibody for those 5 vaccines. So from the left, you have the Moderna messenger RNA vaccine, then the Pfizer messenger RNA vaccine, then in the center, the Novavax protein vaccine, and then the 4th one is the AstraZeneca chimp adenovirus vaccine, and then the last one is the Johnson & Johnson adenovirus vaccine. So note that the neutralizing responses to these vaccines are different. On the y-axis is shown the height of the neutralizing antibody, and you can see that the messenger RNA vaccines produce the highest level, with the Novavax vaccine being second, and the adenovirus vaccines being lower. Now, in the boxes are the vaccine efficacies for each of those vaccines. And you can see that the vaccine efficacy follows the neutralizing titers. In other words, that the highest efficacy is given by the messenger RNA vaccines because they produce the highest level of neutralizing antibodies. And contrariwise, the adenovirus vaccines produce the lowest neutralizing titers and the lowest efficacy against infection. But life is more complicated than that, because T-cell responses are also important in modulating disease, so that the fact that all of these vaccines produce T-cell responses enable those vaccines to largely protect against serious disease, even if infection takes place. Next slide. So the next slide, in a way, sums things up. With respect to correlates of protection by many vaccines, polytheism is preferable to monotheism. And what I mean by that is that multiple immune responses are important for protection. But we must choose the most important one to measure so that we can predict how well a vaccine is going to protect. Next slide. The next slide then reaches my conclusions, which are that the immune system is redundant. There are many immune functions. However, almost all current vaccines work through antibodies in serum or on mucosa that block infection or viremia or bacteremia, and thus provide a mechanistic correlate of protection. But the functional characteristics of antibodies, as well as quantity, are important. And the last slide, next slide, shows that antibody frequently may be highly correlated with protection, or at least synergistic with other functions. That immune memory is a critical correlate because efficacy has to persist for some period of time. And if the correlate of protection decreases, then efficacy will decrease. So effector memory is important for short incubation period diseases, central memory for long incubation period diseases. Cellular immunity may act to kill or suppress intracellular pathogens and may also synergize with antibody. But relatively speaking, most correlates of protection depend on antibody. However, for some vaccines, we have no true mechanistic correlate of protection, but only useful non-mechanistic correlates for an unknown protective response. And I might mention here that rotavirus is one such vaccine where we have actually many different correlates of protection that contribute to the efficacy of rotavirus vaccine. Thank you very much.
Thank you very much for your wonderful talk. We have several questions for Dr. Stanley, but unfortunately, due to the time differences, Dr. Stanley cannot join a live Q&A session, but he already sent his answers to the questions you already sent to him. So I'll show this slide, Q&A slide, to after the talk with Dr. Peter Kwang. So let's please welcome our last speaker, Dr. Peter Kwang. Dr. Peter, please.
Hi, my name is Peter Kwang, and I'm Chief of Structural Biology at the Vaccinology Center at the National Institutes of Health in the United States. And I'm here to speak with you today about structure-based vaccine design. I'm going to share my slides now. Great. So hopefully you can see my slides. The first one is going to be on structure-based design. And I'm going to be breaking up my talk into 3 parts. First, I'm going to be speaking about structure-based design and the Vaccinology Center. Secondly, I'm going to be telling you about paradigms of antibody to vaccine. And finally, I'm going to be saying a worked example of how we're trying to apply these paradigms of structure-based design to develop an effective HIV vaccine. As you know, there's been a long history between structural biology and design because atomic level structures allow organic chemistry to be applied directly to the manipulation of biological interactions. Shown here is a picture of the H3 protease in cyan and green, and there's an inhibitor, protease inhibitor, with a small molecule drug. In order to develop better inhibitors, if you crystallize this molecule together, you can see directly the interactions between the two, and it's just reducing this complex biological interaction to organic chemistry to manipulate and make better interactions between drug and inhibitor. And the success of these types of structure-based design approaches in drugs have led to the question of whether or not you could use the same atomic level design of vaccines. And so that's what I'm going to be addressing today. As you know, you do not need structural information for vaccines. And in fact, shown here in classical vaccinology with Maurice Hilleman, who developed viruses to vaccines for many different pathogens. And his paradigm was to isolate, inactivate, and inject. And he could do so without structural information and developed almost 40 different licensed vaccines, including many, many different pathogens going forwards. However, there are a number of pathogens that resisted that type of isolate, inactivate, inject paradigm. And some of these include like influenza. Shown here is the trimer structure of influenza. It was solved in 1981 by Ian Wilson, John Scahill, and Don Wiley. And when this was published in Nature, it had detailed exact information on the way this central critical part of the influenza virus was being used to infect humans. Couldn't you take this information and use it in vaccine design? Well, not only did we have that structure, but we also had the structure of rhinovirus from Michael Rossman showed in 1998. And rhinovirus is the cause of the common cold. Or you had the structure that I solved in 1998 of the H1G20 shown in red binding to CD4, the human receptor. So shouldn't all this molecular detailed atomic level information be useful in vaccine design? Well, in 1997, then President Clinton gave a speak at Morgan State and said, we need an effective HIV vaccine. And that led to the creation of the Vaccine Research Center, which I was recruited as one of the founding members by Gary Nabel. There were about 10 of the PIs that were recruited at that time. It's a 5-story state-of-the-art building that opened that focuses on HIV as well as many other pathogens, Ebola, influenza, etc. Critically important is that it has its own GMP pilot production plant. And so that means we can go from basic research in this building to creating the mutagens that are needed to create clinical trials and figure out if things will actually work experimentally. John Mascola took over as the director of the center in 2013, and shown here are many of the initial principal investigators that were recruited by Gary in 2001, as well as about 4 or 5 other members that joined subsequently. At the Vaccine Research Center, the organization is set up as follows. First, we do basic research in Building 40. If anything works or is interesting, then we go to vaccine engineering, where we develop the exact processes to make GMP quality products. Once we have that, the technology is transferred to actually the vaccine pilot plant up in Frederick. We actually manufacture GMP quality immunogens that can be tested in humans in clinical trials. We also perform those and then read out the immune responses, human immunology with a Vino, using a lot of robotics and other things. And then once we have the actual results for the clinical trials, we can do more basic research to figure out, do we need to improve immunogens in this iterative improvement cycle? For easy vaccines, when the vaccine immunogen induces highly effective protection, research and clinical trials can start and be completed in just a few months and go from basic science, for example, against Zika, to 6 months, actually more like 3 months later, the actual clinical trial. So very, very fast development. However, if there's no vaccine immunogen has been identified that elicits highly effective protection, then instead of proceeding very rapidly, all the research stalls in basic science. And so the $64,000 question is whether or not structure-based vaccine design can help in this process. That leads to me to the second part of the story, moving from antibody to vaccine. As you know, vaccines in general create very good antibodies, but so do infection by natural pathogens. Dennis Burton came up with this very interesting idea of going from antibody to vaccine. And the idea behind Dennis's thought is that when you're naturally infected, you can find antibodies that oftentimes neutralize effectively the pathogen that you've been infected with. This can be due to virus display or other sequencing technologies, allows us to identify the antibodies responsible for protection. Then working backwards, we can figure what the antibodies recognize by determining the structure with the antigen. And hopefully we can figure out the immune design that led to the original development of that antibody. Dennis pushed his ideas collaboratively with a group of us that formed the neutralizing antibody consortium. And over the last 20 years, we've been working to develop an effective HIV vaccine. In addition to the overall paradigm of going from antibody to vaccine, John Mascola and I at the center have overlaid on top of this informatics philosophy that the more information that we know about the whole process of antibody development, we can use this information in vaccine design. So natural infection normally has the initial nascent immune system, HIV infection occurs, and then that pushes the development of many antibodies to create population effectively neutralizing antibodies that will protect someone who's been infected, or at least control the pathogen. There's a constant cat and mouse game between virus developing an immune system, and so this happens over many years. If we can follow the development of these antibodies through next generation sequencing technologies, we hope to recreate it with different immunogens that will drive similar processes to replicate what happened naturally, get population effective neutralizing antibodies in the vaccine context, so that hopefully they can be used prophylactically to prevent protective humoral immunity. There's a little side part here that's going forwards as well that you can read in some of these reviews, especially shown here in some of these references, where passive antibody can also be delivered to test whether or not antibody can protect. And more recently, we just did the AM study where antibody VRCL1 was shown to actually experimentally protect humans from infection by HIV-1. I'm going to be telling you about 3 antibody to vaccine approaches, and I'm summarizing them here. The first is a confirmation-based approach, and that works for vaccines if a specific conformation of the immunogen can elicit protective antibodies. So you have the whole immunogen. If it doesn't work just straight off with the VLP or the virus, you might have to stabilize one particular conformation of that particular immunogen. And so you have antibodies all over the surface that can work to effectively protect you from that pathogen. Oftentimes that fails, especially for very sequence diverse immunogens, where you might have the immunogen, but you have lots of antibodies, but the antibodies will only protect against one particular pathogen, not very broad ones. And so you might have to focus the immune response to one part of that immunogen, which is going to be epitope with the supersite approach. Sometimes that even fails even further, where you focus the immune response to one site, but even focusing the response to that site might lead to very, very divergent antibodies that are not good. And so instead of that, you could, in an even more extreme case, try to get one neutralizing antibody and replicate that antibody and expand it so you have essentially one type of antibody that gives that immune response. And that's kind of the niche-based design. I'm going to be showing you in detail several of the exact confirmational-based approaches and other things that are going forwards in detail shown schematically here. So first, for the confirmation-based vaccine design with RSV-F, one requirement for this is to have the confirmation antigen that induces antigens that effectively neutralize pathogen. RSV is respiratory syncytial virus, which in the United States is the leading cause of infant hospitalization. And the F-glycoprotein is a fusion machine that's allowed an entry, which has two dominant conformations, pre-F, the conformation that elicits antibodies that effectively neutralize RSV, as well as a post-F that doesn't elicit very good antibodies. For guiding antibodies, a number of them have been isolated. Interestingly enough, synergist and motivizumab used clinically to treat RSV is not a very good guiding antibody because it binds to both prefusion or postfusion as shown here. But we were able to identify very good guiding antibodies that are prefusion specific. Because they were prefusion specific, we could use them to identify the prefusion conformation. And by determining the structures of prefusion and postfusion, which were done by Jason McClellan, who was at that time a postdoc in my group, we could then figure out, well, prefusion, postfusion, what particular modifications could we make to fix prefusion over postfusion? And one of them was to introduce the disulfide from serine 290 and serine 155 and make them cysteines. Now, these are very close to the prefusion conformation, as shown here, but are actually very far away in the postfusion conformation, over 100 angstroms between these two residues in this conformation. So if you link it with the disulfide, you're fixed in the prefusion conformation. We also found cavity-filling mutations that fill holes in the prefusion structure actually help stabilize it even further. And more recently, performed a clinical trial with GMP-produced RSV-F DSCAV1 produced at the Vaccine Research Center clinical pilot plant and tested immunologically as published and shown in this Crank-et-al. Study that was published 2 years ago in Science. It induces 10 to 20-fold higher neutralizing response in humans. And so that's the basis of phase 3 trials now that are being continued on this with GSK and other companies. For the epitope or supersite approach, for the example I'm going to show you is influenza, which is, as you know, quite sequence diverse. If you immunize with the hemagglutinin of influenza, you can get antibodies, but the antibodies will only protect you against the same strain, very, very specific neutralization, not against very, very diverse strains you might encounter. And so we know on hemagglutinin shown here, this is a very conserved stem as highlighted with this red. In this very, very conserved stem, you can have a number of different antibodies from humans that will neutralize very broadly, very diverse influenza viruses. Some of these antibodies are shown down here with different pictures. If you look at the Joyce et al. Paper in 2016, you can see more of those antibodies that have been identified in human clinical studies. Now, how can we take the hemagglutinin and modify it so it induces more of these stem antibodies? One way to do so is to cut off the top. So we can make a headless trimer that Jeffrey Borington did in this paper. And then we can actually place these headless trimers on self-assembling nanoparticles, as shown here in yellow is the ferritin. And this multivalent display gives very good high immunogenicity. You can see the stem now is projected and exposed and gives very good responses. And a clinical trial of this has been started, as shown here in this government study going forward. Finally, there are other cases where you have particular sites of vulnerability where many of the antibodies against it are not broad. In fact, one of those cases was with HIV, and the C4 binding site elicits many antibodies, most of them are non-neutralizing. So how can you, instead of focusing on that site, the idea of that is perhaps we can focus on getting a particular class of antibody. Now, the category of requirement for this is that you need to have an antibody that can effectively protect from the pathogen, and it needs to be listed in the general population. So more recently, we found that a number of antibodies, even though everyone's immune system is different, many people can create what's called VRC1 class antibodies. And in 2013, in this paper by Changqing Zhou, we showed that 9 different donors had antibodies. The antibodies are all shown in blue here, the antibody binding fragments, could bind the C4 binding site, as shown here in red, on GPO20 that's shown in gray. And so that created the basis for potentially what we could do going forwards. For guiding antibodies, what's very important is we can take these sequence class antibodies. Not only do we use the mature antibodies that are oftentimes substantially affinity matured, but we also use the initial germline recombinants, the unmutated germline ancestors, that we can approximate by doing V gene reversions and create these to figure out these are the molecules that need to be trained to induce the B cells going forwards. Bill Schief did a lot of this work and was published in 2 science papers in 2015, 2016 that showed how multivalent presentation of the IO domain could engage some of these germline recombinants and really push and initiate the vaccine going forwards. And more recently in this Duan et al. Paper, we showed how you can take the initial molecules that Bill Schief had created, and you can add glycans, as shown here in orange, to really cover mask most of the surface, so you have much more focus on the C4 binding site. And one example that Bill Schief has pushed forwards with EODGT8 is now being assessed of some really interesting results on the initiation of lineages through the EODGT8 germline priming immunogen. So those are some of the different ways that you can use paradigms of antibody vaccine. And I'm going to be telling you about how we've been using some of this information structure-based design to create an HPV vaccine. Now, for HPV, one of the first steps is to figure out which possible structure-based design paradigms should be used. For confirmation-based approaches, you need to have the entire confirmation be able to elicit something that's neutralizing. And we were able to, and others with John Moore especially, work to stabilize the conformation of HIV into this closed shape, and we figured out disulfides to fix it in that particular shape. However, when we used the conformationally stabilized closed envelope trimers, we could get neutralizing antibodies against tier 2 strains. But the neutralization by each prefusal trimer turned to be very specific to a autologous strain, just like in influenza, where you only get responses against one particular strain that you've been immunized with. So that didn't work so well. We also decided to try epitope focusing. That could be another way where you focused on very particular sites. If a site could be found, they could elicit broad responses. And in parallel, you've just seen some of the work that's being done in VRC01, where if the epitope approach doesn't work, you can potentially do one particular class of antibody, like the VRC01 class. What I'm going to propose is that potentially the epitope approach might be the best approach, because if you can find epitopes here, instead of being choosing one particular lineage, you might not be able to extend so well to really dominate and get protective responses. You could have many different lineage that would work against one particular epitope. So that's what I'm going to tell you about now. First of all, how do we start analyzing all these epitopes? Shown on the right-hand side is a picture of HV envelope sticking out the viral membrane. It's a trimer formed of GP120, GP41. And with these shared epitopes, we can figure out many antibodies that bound to them, including V1, V2 that bound the apex, glycan V3 that bound also towards the apex, C4 binding site that's shown in red, the silent phase, fusion peptide shown in orange, or the subunit interface. And when we analyze all these different features going forwards in this paper that Guayu did in Structure in 2019, we found one of them, VRC34's potential candidate, because it had low glycan content, low number of epitope segments, and high epitope confirmation variation that suggested it could be very good immunogen. Here's a structure of VRC34 binding, and you can see it binds just 8 amino acids primarily at the end terms of the fusion peptide, with 90% of the binding for this antibody is essentially to this linear region. We could take this linear region, take just 8 amino acids, couple in a multivalent fashion to KLH or other types of priming reagents, and then boost with trimer and find very good antibodies, up to 60% breadth in many animals, including non-human primates. Interestingly, our work with non-human primates showed the reason that this worked. If you immunize with trimer only, you get responses all over. And so the antibodies don't know where to focus. They don't know what's conserved or what's variable when you only have a single immunogen. But if you were to prime with the peptide, then antibodies could develop by first binding peptide. You see lots of cytokine mutation with high affinity to this peptide. And then this antibody has the breadth against this peptide conserved region versus lots of other parts that might not be nearly as conserved. We're starting a human clinical trial on this. Here's the schema. there's dose escalation, as well as group 6, which is trauma only, and then 2 prime boost groups, one with the priming with this FP, or FP in context with trauma 4571 is being attempted. And that clinical trial should start Q1 next year. So in summary, I've told you for structure-based vaccine design that structure-based design may enable vaccines to be developed against even the most recalcitrant pathogens. In terms of the antibody vaccine paradigm, structure-based approaches employing conformation, epitope, and lineage paradigms are beginning to show promise in clinical trials. And in terms of our work with HIV, vaccine design based upon the epitope of antibody VRC34, comprising primarily the internalization fusion peptide, is now yielding broad H1 neutralizing antibodies in multiple vaccine test species, and we hope to start experimental medicine trials next year. A few closing remarks on viruses with pandemic potential, because these are also things that are being used for structure-based design. As you know, for the beta coronaviruses, we had the crossover for SARS in 2003 and then MERS in 2012. Barney Graham and Jason McClellan worked on some of these going forwards more recently and showed that you could stabilize in the Paulson paper using 2 proteins at this critical part of the prefusion conformation. You can see in these electromicrographs, MERS wild type shows both post-fusion and prefusion, with the 2P mutations all prefusion. Same for SARS, initial SARS, you can see both post and pre turning to all pre. So Jason McClellan took some of this work very early on when we first heard about SARS-CoV-2, solved the structure, stabilized it as S2P, and this was published in Science in February 2020. And based upon these, as you know, there's been a lot of development with different companies. Operation Warp Speed was started by the US government, choosing 2 companies with nucleic acid, 2 companies with viral vectors, and 2 companies with protein subunit. The nucleic acid have moved very, very quickly, the viral vectors pretty fast, and the protein subunit are still in phase 3 trials. But all of the vaccine immunogens thus far have used the prefusion stabilization such as 2P, except for the AstraZeneca Oxford. So it's a widely used technology, structure-based stabilization. So that's the story for today. Happy to answer questions, and to thank you for listening to my presentation.
Thank you very much, Peter, for wonderful presentation. Now we move on to the Q&A session. Would you please show? Okay. Now you have some questions, and already you have an answer from Stanley and Peter Kwang. So we'll start from Stanley Plotkin. The questions about correlative vaccine-induced immunity. So first question is, how do we determine if humoral or cell-mediated immunity is more important in the efficacy or effectiveness of vaccines? So Stanley answers by statistical evaluation of correlation. In other words, does protection go up with increasing antibodies or with increasing T cells? The latter usually measured by cellular proliferation or cellular killing of infected cells. Neutralization is the function usually measured for antibodies. Now move on to second question is, what could be the outcome of administering 2 different vaccines in terms of immunological memory and protection against COVID-19? Answer from Stanley. So far, the evidence is that mixing vaccines does not decrease protection against COVID-19. At least that appears to be true for the messenger RNA vaccines and the adenovirus vaccines. That being said, it is best to give the first 2 doses of the same vaccine, whereas for the third dose, a different vaccine seems to be just as effective. And so the question, is immune correlative protection established for T-cell responses? And Stanley answers, no. This is an important gap in our knowledge. 2 T-cell functions are commonly measured: proliferation by ELISpot and cytotoxicity. The latter is probably most important. Now we move to the Peter Kwang, structure-based vaccine design. The first question is, how do the vaccines respond to the rapid mutations of COVID-19 strains? And Peter answers, because approval of vaccines may require years, it can be difficult to respond to the rapid spread of mutant strain, such as occurs with the seasonal flu or SARS-CoV-2 variants of concern. One faster response is to use an approvable vaccine as a boost. To increase the immunogenicity in vaccines, this response is faster, since it does not require additional new manufacturing. Second question and the last question. For new technology used as a vaccine antigen, is there any potential risk at cellular or molecular level? Peter says, for new technology such as messenger RNA delivered vaccines or new viral vectors and or new adjuvant, there are clinical risks of adverse responses. Some of these risks derive from hypothetical concern. Other risks derive from clinical observations. Regulatory bodies such as FDA or in the USA balance risk versus benefit. In general, the messenger RNA delivered COVID-19 vaccines appear to induce robust protection with very low incidence of severe adverse responses, which appear to be strongly outweighed by the benefits of protection from COVID-19. This is the last slide for today. Thank you for all presenters today, and thank you all participants for attending this lecture and live Q&A session. See you tomorrow. Goodbye.